Today we are very happy to have our IAS Senior Visiting Fellow, Professor Tom Daniel, from University of Washington, Seattle, to give a distinguished lecture. So Professor Tom Daniel is a Komen Endowed Chair of Biology, and he got very rigorous training in both biology and engineering. And he was actually the founding chair of Department of Biology at UW from 2000 to 2008. And he is also currently the co-director of Washington Institute of Neuroengineering, and the new center director of the Air Force Center of Excellence on Nature-Inspired Flight Technologies, and abbreviated as NIFTY. So if you are interested in knowing what NIFTY is doing, you can do Google NIFTY UW Seattle, and you will find all the interesting assignments they have been doing. And Tom has received several awards, including the recent MacArthur Juniors Award. I think for those of you who know MacArthur Juniors Award, and that speaks the level of Tom's talents in science as well as innovations. He also served in many of the journal editorial boards, including he serving on the editorial board of the Science magazine. And he's also is on the scientific advisory board of Paul Allen's Institute of Brain Science. Many of you know that Paul Allen also, although is very interested in the sports enterprises, and he's also one of the major philanthropists, and he has used his own money to set up several important institutes. And Tom has been in the position to advise various research activities to Mr. Paul Allen. And today we are very pleased to have him here to share with us some of his research in the domain of dynamics of biology. And the title of his lecture is here. Without further ado, please join me to welcome Professor Tom Daniel for this lecture.
It is my pleasure to be here, and I want to begin my lecture with the most important part, which is to acknowledge my thanks to certainly my funding agencies. So if you fall asleep between now and the end of lecture, you need to know that I am very grateful for Microsoft products. Okay, one more time. Thanks, and thanks to my funding agencies. As you'll see, and this is a note to some of you who are doing computational work, Amazon is currently funding students by giving free cloud-based computing. So it's something to know about. And I want to thank Professor Singh for being such a gracious host, BME and IAS for hosting this visit. And I would fail completely if I didn't acknowledge the fantastic students I've had the opportunity to interact with here. The students, particularly in the BME program or the BIEM program, you've been really fun to work with, really exciting, really challenging. So I've thoroughly enjoyed this part of my visit. So hats off to the great students here and the faculty that are recruiting them. And these are my great students who have done really the heavy lifting on this. They took their dress code from Professor Singh, but don't smile as much. I want to draw particular attention to the students with the halos. These are ones whose work I'm really going to focus on today, as well as my collaborator at the Illinois Institute of Technology, Dr. Tom Irving. He's possibly the single world's best expert on X-ray diffraction, and that's in a particular kind that's super critical for this research. So I want to call this out. The students have ranged from graduate students and postdocs to even undergraduates working on this project. All right, let me begin by putting my talk into a bit of perspective. My overarching research interest is control and dynamics of movement in biology. Movement spans everything from the level of sensory information processing to the ability of animals to detect light, feel motion, hear things. All of these sensory acquisition processes are processed centrally We're very interested in central processing of sensory information. And I've had some exciting conversation with faculty here looking at sensory processing, even multimodal processing. That sensory information is processed, decisions are made, and actions are taken and manifest as neural commands of movement. We're very interested in the packaging of information from sensory information to motor commands and what the transforms of those are. It's known in neuroscience as the sensory motor transform. How do you take sensory data and make the appropriate motor? Those motor commands activate muscles, and muscles in turn generate forces, generate torques and deformations. And this is where I'm going to focus most of my talk today. And however, those forces, torques, and deformations generate structural movements. So structures interact with the environment, whether it's a leg or a wing interacting with fluid or a fin interacting with water. All of those structures propel the organism in such a way that those forces change the sensory input. And this is called closed-loop dynamical systems. While my lab is focusing on elements of these, rather than giving you a mini tour of each of those, I've chosen to focus today on just one subtopic of this. And that subtopic is how muscles generate force. It turns out this is something I have been worrying about and thinking about since I was in graduate school, and always have been doing elements of analysis of muscle force. Muscles, as some of you may know, and I will review this, consist of a highly organized, nearly crystalline lattice of proteins. And that crystalline lattice is structured in sort of a scale from centimeters, as you'll see, down to angstrom scale of organization. I'm going to review this with you, but they're dominated by the presence of molecular motors that interact in this highly structured, densely packed, and I really want to point out that there's not a lot of room in there. This is a densely packed array of proteins. And those proteins undergo conformational change at the expense of energy to generate force, and this is what I want to look at today. So what I put in my title was the following is multiscale dynamics. And what we know, and what many molecular biologists point out, many fundamental cell biologists point out, is events at the very tiny scale propagate upward. The events at the angstrom scale and the deformations of the protein propagate upward, and how it binds with other proteins, and how those proteins affect sort of cellular scale events, and how those cellular scale events affect whole muscle scale. But it is also absolutely true that the scale of the whole muscle, when your leg interacts with the ground, forces are generated that in fact determine how those proteins can interact with each other. This is very much itself a two-way street, and this is the multiscale problem in muscle force generation. And it's something I think is becoming more generally the issue in cell biology. In biology is events at the very large scale propagate down to the nanometer scale and outward back again. The other thing I'm going to point out is in cell biology, and in particularly muscle cell biology, things are much more spatially organized than we have been giving credit for. And there isn't much room inside the cell. So what I want to do today is I love history, so I'm sorry, I'm going to give a little history lesson about our understanding of muscle, because I just find it interesting. Okay? So you're going to, and those of you who have suffered through my lectures earlier this week, you know I love old science, and so I'm just going to review some. And just to get everybody on the same page, I need to go over the micro and nanometer scale geometry of muscle, so we understand how the players are arranged and how they interact. I'm then going to step into something that we began about 20 years ago as a series of molecular motor modeling problems. And what we kept running into is, how do you account for some very complex dynamics in this very structured intracellular environment? And then I'm going to focus then on how we can actually get data, measurements that relate these models to reality, As one of my colleagues said, models are doomed to succeed. And finally, I want to conclude my talk not with everything we have learned, but much more about where we think there's some problems and where we're unsure of where to go next. Okay? So I'll give you the mini history of muscle now. To records that I can find, it begins somewhere around 180 BC to the physician Galen. And Galen identified in neurons, nerves, that he thought that the spirits of animals move through nerves and that there's a movement of humors, not a humorous movement, but a movement of humors, that's fluids, or some spirits moving in nerves. And in fact, he would cut nerves and note that motion would disappear. So clearly this was support of his idea, scientific idea, that humors or something is moving in the nerves. Now actually, at the end of the day, he's not fundamentally wrong. Things are propagating along neurons. They happen to be action potentials, which are not humors or even humorous. Around the time of what we call the great anatomists, this is Valverde and Leonardo da Vinci, they looked to muscle and its organization as a structural issue, as a geometric issue, as a functional device. And in fact, in da Vinci's work, if you look carefully at many of his drawings and studies, he begins to think of muscles as force-generating devices that produce lines of action. I quoted an earlier Leonardo quote for the group Nature Inspired Flight, where he identified Bernoulli's principle 200 years before Bernoulli. It was absolutely stunning insight, this gentleman. So we have some sort of structural analysis of muscle, and we have the humors, and these were brought into focus by Descartes. And he actually gave a mechanical hypothesis to the idea of humors. And the mechanical hypothesis was that when, and I have a quote in my notes, but I'm going to do my best to reproduce what Descartes said. He said that, the fire here it moves very quickly and it touches this toe and it runs that feeling up this device here, and this big empty chamber, which happens to be one of the ventricles in the brain, which contain fluid, he thought would pump fluid down into the muscle to get it to contract and pull back very quickly. So the feeling drove a hydraulic sense of how muscle works. Shortly around that time, a Dutch scientist named Swammerdam decided to prove that Descartes was right. And what he did is he took muscle, and he placed it in a liquid chamber, had a wax covering, and brought the nerve out, right? And he had in this chamber of fluid a little bubble. So his point was to show that when he squeezed on this nerve, and it turned out it would get the muscle to contract, as you'll see shortly, that he was expecting fluid or some volume change to happen. And so he was going to show that this bubble here would move up and down as the muscle contracts and changes volume, like this hydraulic idea of Descartes. And his writings go on to show how absolutely depressed he was, that the muscle did not change its volume. Needless to say, he was somewhat deflated in his view of this. So Swammerdam went to show that Descartes' idea of muscle volume changes was correct, and he left this whole process depressed. But we all know muscle is made up of cells. Cells actually don't, on short time scale, change volume. Okay? How many agree with what I just said? A bunch of students in here. Are you free to raise hands here? How many agree that cells in short time don't change volume? How many disagree? Okay. How many have no opinion whatsoever? Okay, that's right. Most of you agree. I'm okay with that. The reason I ask that is this is going to come back and haunt us today. right? And that's why I'm bringing this up. This idea that if you touch the nerve actually was verified by Galvani, where his assistant, and I talked about this earlier to the class, his assistant, keep hitting the wrong button, his assistant touched the nerve of the frog with his metal tools, and the frog leg jumped. And this is on like half a frog, so there's no brain. And so Galvani and his assistant suggested that it wasn't humors, there was electricity. The electric spirits moved from his assistant into the muscle and drove the contraction. Volta later showed that in fact was not true, that the metal of the tool interacted with the salt solution around the nerve and created an electrical potential, much like a battery operates. And that's hence known as the Voltaic reaction. This idea of stimulating muscles was used in I would say, the first hardcore case of bioengineering, which was Guillaume Duchenne in 1850s, showed that if you stimulated just electrically the muscle of an amputated arm, you can get the fingers to move. By the way, this was totally creepy to people at the time. Electrical stimulation would get muscles to move. And in fact, Mary Beth Shelley noted that this was like reanimating dead things. An arm is cut off. What if you did it to a whole body and wrote a book about it, and it was called Frankenstein, the monster? Reanimated by electricity. Electricity was the molecular biology of that era. It was the hot topic, right? And so everything was getting electrified, and people were getting shocked, and Duchenne was certainly having a good time doing it. What he noted is that you could use electrical stimulation of muscle as a diagnostic tool for individuals that have a family inherited disease called muscular dystrophy. It is now one of the forms is called dystrophin, Duchenne's muscular dystrophy, named after this. And he used this as a diagnostic tool. Enough of the distant past. I want to sort of wrap this up now with who I think is one of my heroes, is Andrew Huxley. And Huxley, as many of you know, won the Nobel Prize and did so largely for his understanding and resolution of the molecular mechanism by which nerves conduct electrical signals, actually the molecular and protein basis of the action potential. And if you stop and think about why he won the Nobel Prize, it wasn't that it was just grinding work. They had to have some deep insight into positing the existence of a protein behavior that is a voltage-gated behavior or a voltage-dependent conductance of a protein. They had to posit that existence in a theory in order for it to explain the propagation of action potential. They had to posit the existence of something not known. That, to me, was phenomenal, and how that insight was reached. There was precedent for it, but then they went on further to actually do the measurements. And that was done in the squid giant axon. But equally important, and to me, possibly just as worthy of the Nobel Prize, was his understanding and theories of how muscles generate force. Just as he had to posit the existence of a protein that changes conductance, ionic conductance, in a voltage-dependent manner, he had to posit the existence of a protein that converts chemical energy into mechanical work and does so in a powerful and effective manner in force generation of muscle. That became known as the sliding filament hypothesis of muscle contraction. Okay? And this is what I want to focus on now for the rest of the lecture, which is the sliding filament hypothesis. And I will preface everything by saying that when I said sliding filament, the image, as you'll see, is of filaments sliding past one another, and we'll explain that in a minute. And I just have this quote from the obituary in the New York Times. His boyhood mechanical skills led to a career in physiology. And the translation in Huxley's mind was the mechanical engineering of living things. That was a quote from Huxley. And he won the Nobel Prize for explaining the electrical basis of bodily movement. And there is some interesting biography about why he thinks actually the muscle one was more interesting. Okay, let me now get us on one page about the geometry of muscle. And many of you know this, but I'm still going to review it. Muscle, as you know, is a large multicellular tissue, highly organized. In that tissue is made up of muscle fascicles, and those fascicles are made up of single cells. They are large cells, huge cells, that are syncytia of subcellular elements. These are called myofibers, and those subcellular elements are highly organized structures running longitudinally called myofibrils. Don't worry about the names. And those myofibrils, if we look very closely, have a very, very regular, highly crystalline structure. That is, there's very thin filamentous proteins extending inward and very thick filamentous proteins interdigitating with them. And for once, biology has a decency of naming these logically. The thin filamentous proteins are called thin filaments, and the thicker ones are called thick filaments. I thought that was useful. And the thin filaments interdigitate with the thick filaments, and these motor molecules, made predominantly of the enzyme myosin, myosin 2, conventional myosin, interacts with these thin filaments made predominantly of the protein actin. Okay? And it's that interaction I want to explain today. So we're diving down, all the way down. We're right now at the sort of nanometer scale, okay? And that nanometer scale, I'm going to show you blow up of those proteins, has the following interaction. There's this structure here. There's the myosin from a Rasmussen plot. And this interacts with ATP and ADP. ATP, in this case, it's ATP with a phosphate and ADP. They interact with this thin filament. Let me go over this interaction. And I apologize, but this is just to kind of get the machine down. Okay? So the thinking is, and this is a fairly simplified view, okay? The thinking is that we have this enzyme that hydrolyzes ATP into ADP, adenosine diphosphate, and phosphate. And that hydrolysis, upon the hydrolysis, the space occupied by ADP and phosphate, that associated phosphate, is actually much larger than the space occupied by ATP because of that hydrolysis. And it's like you took that ATP molecule, hydrolyzed it, and you expanded a particular region. The molecule itself has some elasticity. Okay? All structures do. And so that expanded region has now stored elastic strain energy in the molecule in a particular region around a deforming region of that molecule. When that molecule in that hydrolyzed state attaches to the thin filament, and it doesn't reach out and grab it. It diffuses, an elastically bound structure subject to thermal fluctuation that diffuses to a binding site. And upon diffusion, when it attaches here, there's a steric change that phosphate drops off. And now that space occupied by that spare phosphate is gone, and it's like you pulled the lever out of a spring and bang, that molecule can undergo this conformational change. And importantly, ADP and ATP, yes, there's that phosphate. They're not as big a spatial difference as there is with ADP plus phosphate. And so ATP can come in and substitute for that ADP, and you get that release of the molecule, okay? And that release, that substitution of ATP for ADP, resets that molecule, and it's ready for, it hydrolyzes that ATP into adenosine diphosphate and phosphate, and then it's ready for subsequent cycles, and that goes on. And if you're like me, a movie is worth what I just said. So here's a short little animation. So the phosphate falls off, that molecule undergoes some sort of stretching along here. This is animation, this is not, and some twisting here. That ATP is substituted for ADP, the molecule releases, you hydrolyze, reset, the spring, bind, and regenerate. So this is cyclic conversion between hydrolysis of ATP, right, and a mechanical strain energy stored in the molecule. That repeats over and over again. Great. And all of that happens in three-dimensional, complex, tightly spaced lattice. Okay? And what I've done here is just showed you there's a thick filament. All right, that makes me dizzy. There's a thick filament here with these cross bridges or myosin molecules, the motor molecules, radiating out. It's a 3-start helix that goes up and just continues on through the length of that molecule. And it's in most muscle, but it's certainly not all. It has 6 thin filaments surrounding this 3-start helix. Okay? Distances are absolutely critical. This is an elastically bound molecule. It's about 5 nanometers in head diameter. And it's subject to thermal fluctuations, right? And those thermal fluctuations may eventually bring it to, within, let's say this is a binding site right here, and this is the molecule. Let's say it brings it into reasonable distance of the binding site, and then it can attach, right? And depending on the stiffness of the spring of that molecule, the distribution of locations it can have can be very big or very small. And among all myosins, there's amazing differences of their mechanics. But interestingly, there's along a thin filament, the myosin molecules, here's a set that are all collinear. They face this one thin filament. They are spaced collinearly about every 43 nanometers. And in and of itself, it's not an interesting number. The more interesting number is that the binding sites are every 37 nanometers. Okay? So there's a misalignment between the motor molecules and the binding sites. And importantly, the motor molecules aren't likely to fuse more than 1 to 2 to 3 nanometers, just under the normal thermal fluctuation. So the fraction that are actually locally well aligned is quite small. Okay? Very interesting issue. We got into a lot of debate. This point actually had escaped the attention of the literature, and we use this now in a homework problem in our bioengineering class, is if you have to diffuse to within 2 to 3 nanometers, what fraction of those motor molecules could possibly be bound at any one time? It's an interesting problem. So distances are critical. The motor molecules have to diffuse to a binding site, and they're misaligned. I like the fact that 43 nanometers and 37 nanometers are both prime numbers. I don't know why I like that, but they are. So there's no integer matching. Interestingly, in other structures called cilia, there's a different motor protein called dynein, and it binds to microtubules, which have tubulin, and those are integer multiples separated, 8 nanometers apart for the tubulins, and 24 nanometers apart for the dyneins. Just an interesting issue. ATP and ADP do not get produced in this lattice of proteins. They're produced outside the lattice. There are no mitochondria in the lattice. So there has to be diffusion into and out of this system. Early analysis said that's not a problem because this spatial scale is only a few micrometers. You should be able to diffuse ATP rather easily if this is water. But if it's solid protein, ATP will not diffuse at all. So there's actually an uncertain diffusivity of ATP in this space. Okay? Motion in general is in a cramped space. So I will tell you that the field of muscle biology has not talked very much the field of fluid dynamics. If you have very tight spacing of proteins moving past each other, and the gap distances are tiny, traditional fluid dynamics, we call low Reynolds number fluid dynamics, will suggest that this is very, very expensive. That may not, in fact, be a valid assumption that goes with what are called low Reynolds number. I'm going to come back to these issues. And finally, I'm going to come back to Swammerdam. Poor frustrated Swammerdam. He said, muscle doesn't change volume, right? So as it shortens, what has to happen? As the muscle shortens, yes, I heard it. Yes, what's it have to do? Yes, it doesn't change volume, it gets fatter. Right? And I'll demonstrate that. No, I won't. Let's do so as it gets shorter, I'm not very good at this. It gets fatter, right? And in fact, if the volume is conserved, as you get shorter, you have to get fatter. This is a trivial point, but in fact, that becomes critical. The reason I'm asking that is, do filaments just slide? Okay, think about this. The muscle's getting shorter. Do they spread apart? Do they not? What's going on? How do the cross bridges interact in both the axial direction as well as in the radial direction? We haven't even worried about radial direction. And importantly, one of the other historic figures I brought up was Duchenne and his analysis of muscular dystrophy. Muscular dystrophy is a disease in which you do not produce appropriately the protein dystrophin. Dystrophin is a protein associated with the radial connection of the filament lattice to the surface of the cell. Okay? Interesting. What are the implications of this sort of conservation of mass, conservation of volume to disease state, force, energy storage, energy delivery, even the availability of ATP in the lattice? Okay, so just a brief, quick summary. We did a little mini history of muscle from Descartes to Huxley with Duchenne in the middle, okay? And then muscle, as I now pointed out, is these thick and thin filaments interdigitate with little protein motors that walk one past the other. And this occurs in a complex three-dimensional lattice at the expense of ATP energy. And so now I'm going to tell you about some early modeling that started actually with Huxley towards the end of his career in the late 70s. And Huxley developed what was called the three-state model that I've shown you here. Okay? And he said, let's approach this like I would any sort of biochemical system. I'm going to develop what is called a flux conservative equation. That is, the occupancy in any one state, the sum of all occupancies is conserved. So let me explain that a little more. Imagine there are three states, state 1, state 2, state 3, unbound, weakly bound, strongly bound. Okay? Here, when it binds that phosphate, it's weakly bound, the phosphate falls off, you get that strong deformation, and that then with the ATP substitution. You can then develop a differential equation. This is standard mass action model. It says that the fraction occupying, the rate of change of those occupying state 1 is the rate into state 1 from N2, the rate into state 1 from state 3, and the rate out from state 1 to each of those states. And you can do this, obviously, for all other states. And the really interesting one is what fraction are actually generating force, right? And so that's really the final state, this state 3. That's this state here. And those rates depend on location. So here, I'm just going to give you verbally and not mathematically how we think about that. Clearly, the rate going from the unbound state to the weakly bound state, the first attachment is like a diffusion process. It's actually an Ornstein-Uhlenbeck process in which this elastically bound molecule diffuses to a binding site. Okay? The farther away the binding site is, the harder and less likely it is that that molecule will attach. The stiffer the molecule is, the more rigidly it attaches, the harder it is for it to bind. Okay? The more compliant the molecule is, the weaker the force it will generate when it binds, but the more likely it could bind. So if it's just loose and completely unbound, it could eventually find a binding site, but it will generate zero force. If it's bound with titanium, right, attached by titanium, it won't reach a binding site, very likely. But if it ever did, it would be a lot of force, okay? The other thing is when the molecule is bound, the higher the distortion, the more energy you have stored in the molecule, therefore your rate off has to increase. And if I pull on it, I'm going to increase that energy stored, right? Energy imparted. What Huxley then said is, well, if I have a decent model and I know these rates, and this is really a hard problem, how you know those rates, okay? Says if I know the fraction in that state, that's the fraction there, and I know how distorted they all are in that state, which is I just average all possible distortions, which is from 0 to the largest imagined number that you can get. Clearly, the very, very, very large distortions have very low occupancy. Very small distortions have very high occupancy. And their spring constant K gives you a force. K times the distortion times the fraction with that distortion integrated over all distortions gives you the force generated by the molecules. And then Huxley did something truly brilliant. How could I actually measure this? How could I know the rates? And Huxley did an experiment that published in Nature in 1971. He took a muscle, activated it, but held it, right? Just holding it. And it wasn't a whole muscle, it was a cell, or a few cells. And then he abruptly released it. And so all those cross bridges which were pulling, all of a sudden go, whoa, and binding sites are gone. And so they have to look around, get the next binding site and regenerate force. His idea was an extremely rapid perturbation. All of the mechanical transients that follow that are manifestations of the regeneration of force, the reattachment, and the redevelopment. It's a good idea, but I know there's some physicists in the audience, and I'm going to ask you to help me with something because there's a problem with it. But I'm going to leave that for now. Mass action kinetics works very well if, when one molecule binds, it does not in any substantive way affect the probability of another one binding. Okay? Thus, if I bind to Professor Singh here, and I pull, but he doesn't move, I don't drag Professor Shi closer to me here, right? But if when I bind on Professor Singh and I pull him over and Professor Shi is dragged closer to my binding site, then this likelihood is affecting the likelihood of others, and they're not uncoupled. They're coupled systems, not purely mass action. So the events that go on here are not independent of the events going on in the neighboring molecule or the neighboring one. So there's at least the possibility of coupling. And you could say, well, if I hold my muscle and don't let it shorten, then there's no motion, so there's no problem, right? That would be true, except a series of papers came out in the late 90s by another Huxley, Hugh Huxley, not related to Andrew. He said, you know, proteins aren't titanium. They're not infinitely stiff. And in fact, he showed that there's a considerable elasticity in the thin filaments. And as it turns out, there's considerable elasticity in the thick filaments. It's like strings of springs, right? So now if I bind, even if Professor Avi is not free to move, if there's a bunch of springs connecting them, I can and he still doesn't move, these two can, right? So even in the fixed muscle prep case. That was for the students. I'm sure all the faculty knew that, right? So we began a series of modeling papers in the 90s. And it began very simply by saying, imagine that each motor molecule, like this, is bound to the thick filament by a spring. The motor molecule itself has some measurable and known elasticity. And by the way, it has measurable and known elasticity. And I'll go on that in a minute. And imagine also that this thick filament is a series of springs, and the thin filament itself is a series of springs. And then what I do is I play the same game, the same analysis that Huxley did of a three-state model, but I do it as an individual-based model over and over and over again. And they're coupled by elastic springs, okay? And so all I know is that the forces here all sum to zero at all instants in time. I'm going to ignore acceleration effects at this tiny scale. And we do a force balance. And this is a linear system of equations for, as it turns out, as many proteins as I care to establish. And then I do the same trick that Huxley did, except I do this by a very individual-based model where binding rates depend on distances. I use a Monte Carlo type simulation for each event. Although it's possible to derive a PDE for it, it's just easier in this case because it turned out not to be linear. And we simulate this, and I think I'm just going to show you the results. We ask basically, how do the forces vary as a function of the design of the protein lattice? And this plot here, and let me work you through it, is this axis, the height, is how much force per ATP you generate in a system of molecules. This axis here is how stiff is your thin filament, from very stiff to very compliant. And this axis here is how stiff is your cross bridge from very compliant to very stiff, okay? And what you find is you have this weird surface behavior. And let me go back through this. A very compliant thin filament and a very compliant cross bridge, while your binding probability is very high, right? I have this really floppy cross bridge. You can easily bind to any binding site because it can easily diffuse. Once it's bound there, it's so floppy it can't generate force, right? Over here, I have a very stiff cross bridge, right? And a very stiff filament lattice. So if this cross bridge ever does bind, it would generate a lot of force. But in fact, its probability of binding is so low that I rarely get anything. And right in the middle, I have this situation where the filaments stretch, recruit cross bridges that were not so well aligned, right? Just as the example I showed with our wise colleagues here. And as such, you get this compliant realignment of cross bridges. And you may wonder where in the literature do the data support things? Somewhere right about there. That is, the cross bridges have about 5 piconewton per nanometer stiffness as measured by optical tweezers, right? And thin filaments, as measured also by optical tweezers, have a spring stiffness somewhere around 200 to several hundred piconewtons per nanometer. Okay, all this is fine. We did a whole series of papers. There are some problems. Our observation is molecular motors bind cooperatively because of stretching of molecular springs. Again, I'll just repeat myself. That is, when one motor molecule binds, it distorts the filament lattice to create favorable binding environments for others that were otherwise unavailable to bind. The spring constants that come from the literature, we did not measure these. So these are independently measured, reasonably capture this kind of behavior. It suggests that this is sort of a tuned system. By the way, motor molecules, even in myosins, vary in the animal kingdom quite profoundly. And so there are wide ranges of stiffnesses, both thick and thin filaments, as well as the motor molecule. But these simulations I did were fixed. I held the ends. I just did what Huxley did, but I recruited everything, right? I didn't allow it to change length. And as filaments change length, as the muscle shortens, these filaments change their amount of overlap, right? In fact, there was a classic experiment done by Albert Gordon, Hugh Huxley, and John Julian on what they said was the experimental evidence of the sliding filament hypothesis is they took muscle at different lengths, stimulated it, measured the force, and they showed that at very long lengths, when the muscle's really stretched out, very few cross bridges are interacting, so you get very little force. And as the length shortens, you get more and more recruitment of cross bridges to a point where you maximally overlap. That's as many as you're ever going to get. And then the thin filaments start to overlay each other and begin to hide binding sites. So you get a reduction in force. As they go like this, the thin filaments, that's the idea. Okay? So this was heralded as physiological evidence of the sliding filament hypothesis. Very, very classic result. But as muscle shortens, it's got to get fatter, I think, unless water's shooting out of cells, which I doubt. Okay? So it's possible that the filament lattice, as the muscle gets shorter, has to dilate. In other words, as the muscle goes like this, it doesn't just slide, it would slide and expand. Think about that. If the muscle filament slides and expands, the distance from the cross bridge to the binding site has to increase. And thus, the binding likelihood has to decrease. That's fine. And that would suggest as muscle gets shorter, this should fall. It's a different explanation for the same phenomenon, which has been measured countless times, right? Well, this is where Dave Williams, currently a postdoc in the lab, he was a graduate student who worked on this, then went to Harvard to integrate this into whole scale models, and then has come back to the UW. he did a model of the following. He said, well, you know, first of all, if you're going to start to worry about forces not just in the axial direction, but the radial direction, the myosin molecule has interesting domains that not just stretch, but also undergo torsion. Okay? And so he built a slightly different model in which the myosin molecules are modeled actually as torsional springs with elastic springs. And that's a great idea, but this is not a linear system anymore because these angles introduce a transcendental function, so he can't develop an invertible matrix. But he simulated this as the same kind of problem, an individual-based model. He did so using a Monte Carlo simulation, and then did this for a full three-dimensional geometry of the filament lattice of motor proteins interacting with thin filaments, extending into and out of the board for the correct number of motor proteins, the correct arrangement. And then he had appropriate boundary conditions that fundamentally these are toroidal boundary conditions so he can map everything to the other edge. Okay. And here's what he did, same problem. What he can do is he can specify the overlap and the lattice spacing. How far apart are they this way? How much overlap of filaments? And just specify that, right? And he can also specify the mechanical properties of the filaments, just as we did, it's just parameters, and also similar idea of the cross-bridge kinetic cycle, right? He runs the Monte Carlo simulation. This is all done on AWS, Amazon Web Services, and it was all free, so, which is really wonderful. And he has to undergo a really complex optimization method, but eventually settles everything out to get all the spring distortions, forces, locations, and does so with stochastic simulation. And here are, this is horrible, this is his thesis in a single graph. But this is the lattice spacing, so how far apart the filaments are this way. And then this is the filament sarcomere length, so how much overlap there is this way. And what he finds is that if you have constant lattice spacing, you're just doing the sliding filament problem, you get exactly or roughly, not exactly, you get roughly what Gordon, Huxley, and Julian found experimentally. It's really nice. And if you do the other direction, that is, I'm going to hold the length constant, I'm just going to swell muscle fiber mathematically to spread the filaments apart, he gets pretty much the same result for the same reason. One of the debates we had is why would you get lower force when the filaments are so compressed? And it all follows from the fact that there's an angle change of the molecule to generate force. As the filaments get compressed, the angle that you can accept for binding gets shallower, so it can't generate force in the correct direction. The reality is somewhere probably a combination of these, or so we think. So our rough idea is as muscle shortens, it should follow the constant volume and constant volume approach of lattice spacing changing and filament overlap changing. Get a much more interesting behavior. So what really happens? This was great. So what we're saying is if you change how far apart these filaments are this way, or how much overlap there is, you're going to change force. But what really, really happens? So here I'm going to give you a hypothesis. I didn't say I'm going to prove it. I'm just giving you a hypothesis. The hypothesis is as muscle shortens, the lattice spacing should increase. In fact, it should increase with the square root of the length because the area has to expand as the length changes, right? So the radial dimensions change. Does the lattice change the predicted changes? If the answer is yes, cross bridges would need to reach to a greater distance for actin binding. That is, as the muscle shortens, they have to reach farther and farther apart. Okay? The answer is no. If the lattice spacing doesn't actually change as the muscle shortens, fluid has to be getting squeezed from one compartment of the cell into another. Okay? I don't think I have any other option because I don't believe the density of the fluid is going to change and I don't believe it's going to magically disappear. So, simple math with a less than simple method. what we ended up doing is that we wanted to do sinusoidal length change of contracting muscle, as you'll see why in a minute. We want the muscle to be fully intact. Most experiments now, today, on isolated muscle, they take the membrane off the cell so that they can get the right chemicals into and out of the cell during experiments called the demembranated or skin fiber preparation. So you can control the calcium, you can control the ATP levels, you can control everything. Right? But the problem is when you take the membrane off the cell, you can no longer assume it's a constant volume cell since you've just taken the border off. So we needed an intact muscle preparation, and we needed to be able to lengthen and shorten it and control that. And we want to be able to turn it on at any time, at any part in that lengthening and shortening cycle. So I might want to turn it on when it's lengthening, or I might want to turn it on when it's shortening. In other words, I might want to recruit cross bridges at will, right? And finally, I want to measure in real time, while I'm doing this to a muscle, the lattice spacing in real time at the nanometer scale. And I may as well measure force and length, and I can calculate total energy while I'm doing that. We used our model system, the Hawk Moth, and it turns out it was purely accidental why we ended up using this. Happened to be we have them. It happened to be I was doing some other work on their structures and stuck them somewhere. You'll see why. The most important thing is these muscles have what's called a one-for-one correspondence between muscle contraction and electrical activation. Ours do not. Frog muscles do not. Frog muscles can twitch, but they don't maximally twitch. You have to crank up your frequency of stimulation. These muscles are all or none, one-for-one stimulation. That means I can turn them on explicitly and repeatedly at any point in time. We took our moth, flew to the Argonne labs at Chicago, and built the following. The moth went in a suitcase, so we didn't fly the moth. Somebody was chuckling. Yeah, no, we flew the moth in an airplane. So we took the wings and the head and the legs off. I'm sorry, this is the remnant of a moth, okay? But it still has energy storage, and we isolated the flight muscles, and these are attached to a length driver, a force beam, and in fact, we have electrodes, which are not shown here, that can stimulate the muscle at any phase. Okay, so we're now got the muscle, we can do this, measure force, measure length, stimulate, completely controlling the muscle, just like Duchenne did, just like Galvani did. We take that and we place that in the beam of the X-ray at Argonne labs. And this is let me go back a slide. This beam, the intensities are related to the Fourier distribution, the Fourier transform, the spatial distribution of the muscles. So that what you're measuring is protein spacing changes, right? And it turns out you can measure two directions, because this is a beam going through muscle. So if the proteins spread apart, the periodicity changes and dots associated with that compress in one axis. And as the muscles stretch, the helices change their dimension. So you actually get two deformation dimensions at once. And the reason this works is small angle X-ray scattering has just the right energy level for getting these dimensions in real time. And it turns out it just came online, a shutter speed of an X-ray detector of incredible capacity. And so we synced the shutter to the stimulus of the muscle, right? And so now we're timing everything. And we get these beautiful timed images at, in this case, every 4 milliseconds. So this is super high temporal resolution muscle, X-ray diffraction. And I will end by saying we've gone way past this. I'll show you something at the end. And then we repeat this length change because it's cyclically timed. We can just do 100 cycles of oscillation, and at 25 hertz, that's no time at all, right? And then we take every 4 milliseconds and add it to the one there, take that one and add it, and we phase average, so we get amazing signal clarity of the spacings. And we get a pattern that only an X-ray person can love, but let me work you through it. These two dots here represent the spacing in this direction. As the dots move inward, the spacing moves outward, and it's the Fourier transform. As the dots move outward, the lattice compresses. And we actually get a real-time movie of this. The spacing is actually given by Bragg's law, so we can actually calculate the actual in vivo spacing in real time. And there's a movie of contracting muscle. And we now will go to the cool part of the data analytics. I realize I'm running in on time. We just quickly move on. Here's what the length of the muscle is in one cycle. And so I'm just showing you it starts short, goes long, gets short. Okay, and this repeats over and over. So I just showed you a single cycle. Here is what we would predict the spacing is if it's constant volume. That is, as the muscle is very long, we expect the spacing to be short. As the muscle is short, we expect the spacing to be out. These numbers, these are not measured. These are just resolved from these measured numbers. And here's what actually happened. Neither. Okay, it was neither constant lattice spacing, nor was it purely tracking constant volume. What's important to see here is the muscle was sort of tracking constant volume, and then I turned the muscle on electrically here. And then I recruit cross bridges. They bind and eventually let go, and I come back. It's as if while I'm pulling on the muscle, when the cross bridges bind, they brace the lattice. Well, that's a nice idea, but the whole point is I said I could phase control this. If that's right, and I change the timing of when I recruit the cross bridges, I should change that pattern. In other words, if I recruit my cross bridges here or here, I should see it, brace it at different parts. And sure enough, when I recruited my cross bridges here, there was no change in lattice spacing. It barely tracked. And then when they're off, it's tracking fairly well. Cross bridges are doing something interesting. They're bracing the lattice. But now remember what I said earlier is if the filament lattice doesn't track that isovolumetric assumption, fluid has to move in and out of the lattice spacing. And if it does track it, then cross bridges have to bind to greater distances. But by bracing the lattice, they actually favor other cross bridges to bind, as opposed to letting it go out. They also store energy in a radial direction. If there's anyone in the audience that has a little bit of fluid mechanics background, there should be a little bit of a worry here. That is, I have this intensely packed porous spacing of filaments. In order for the cross bridges to hold that lattice while things are shortening, fluid has to move out of this space. Fluid moving in very tightly spaces generates viscous shears, to our knowledge, which could be energetically very expensive. So the answer to our hypothesis was yes and no. I'm sorry. I thought it was going to be cleaner. It isn't. And so now this part is near the end of my talk. So this is complete rampant. This is as far as we've gotten. We're continuing experiments, and I'm going to show you a few of where we're going. But let me now do a little bit of rampant speculation and welcoming ideas and collaboration. So cross bridges are restricting radial motion, and they do have a role in energy storage. We were able to show this in a whole separate set of papers. And as I said, there could be viscous dissipation of energy. This is a bad thing because what you want to do when you're contracting muscle, you want to generate force. You don't want to dissipate energy. You want your mechanical energy to turn into motion. I also said there's an uncertain availability of this substrate of energy, ATP, into the lattice spacing and getting ADP back out. Is it possible that this restriction of motion not only helps recruit additional cross bridges, it's kind of like breathing, that you're actually using, it's a convective dynamic of moving ATP and ADP in and out of the lattice, which diffusion may not be sufficient for. You get a time framing, a time base of movement of ATP and ADP that may be pumped, in a sense, by this weird interaction of the filaments and the cross bridges. That is rampant speculation. I actually don't know how to show that. It's a hard thing to test that hypothesis, so I'm open. And just to give you a sense of this, this is a cross-section muscle. There's basically, it's completely packed with filament proteins here. And this one has been a fiber that's been expanded a bit. I want to come back to one more thing. I can't let this go. Huxley did this really brilliant experiment where he took the muscle, activated it, and abruptly released it. Okay? Really abruptly released it. And ever since then, we've been trying, the community, has been trying to release muscles faster and faster to capture more and more of the early state kinetics. Right? There are a couple of concerns I have here. And again, I will confess, I wrote a paper when I was an assistant professor with this concern. And I haven't let it go. It hasn't gotten published yet. Huxley reviewed it. He did not like it. Okay? And I kept the review. I said, wait a minute. I was trained in engineering. I know if I take an elastic structure, like a slinky, and I go, pa-town, I get elastic strain waves. It's easy. Anybody can get elastic strain waves. You won't get elastic strain waves, be able to measure them if the stiffness is so high and the mass so effectively low that it's basically like a rod moving back and forth. But I can tell you, if I took a rod and I hit a hammer on it, that strain wave will move at the speed of elastic strain waves based on the modulus of that material. Muscle is an active material. It's an active, very odd energy utilizing material. I cannot tell you what the wave speed is in muscle. I can't hit it with a hammer. Well, I can hit it with a hammer. But it's a hard problem because you have dynamics interacting. So that's a neat physics problem, right? But we can do some measurements. So let's hit it with the hammer. And what we just did, and this is just a few months ago, and I know this is hard to see. It's actually important that it's hard to see. This is our length change. This is the force and some transients that happen afterwards. This little line here is the width of the timing of an image we took, 40 microseconds long. And these dots are the photons captured through the diffraction. And it's kind of hard to see. We cannot do the phase averaging we did with the repeated cycles, right? I go bang, I got to measure. Dave Williams was recruited back to the University of Washington in the data science program to work with some astronomers who have the same problem of identifying star clusters. Okay? Both of us are working with rare capture of photons. Theirs are maybe 40 kilolight years, ours are maybe 40 nanometers, right? But still, what's a few photons between friends? Dave worked with the data science group to do really a neat Monte Carlo Bayesian-based peak detector that's automated. And I need to point out that until Dave did this, the way we analyzed X-ray diffraction is hand digitizing the images. And when you're starting to get images at thousands per second, that becomes an undergraduate's nightmare. So this image is the refined image that we've got. We can actually get post hoc, real-time spacing. There's much more than spacing in this image. And what we're very interested in is that the ratio of intensities, this distant peak here and this peak here, measures the mass distributions, not just the spacing, but how mass is distributed spatially of these proteins in the muscle, the radial direction. We can get both radial spacing and radial mass geometry. Kind of a fun problem. Okay, I've droned on long enough. This is where we are now. Our current research is asking, are elastic strain waves moving? Number one. Number two is it possible that you have this sort of weeping or pumping of fluid into and out of the lattice? And what might be the biological consequences of that motion in terms of energy delivery of ATP and energy storage elastically? With that, I'll just say thanks again to all of you. And I'm happy to entertain some questions.
Very interesting. Appreciate it. I think I wonder. You have the spring torsion system, the top of fluid, but your model does not incorporate the shear force into this.
Absolutely.
I think it's not just pumping fluid in and out. That is not. I think that is not probably sensible from the physical model construction viewpoint, but the surface force
It's huge. or so we think. So I'm going to rephrase Professor Shi's question here. Well, he is correct. You have ignored the fluid dynamics intracellularly. And there are a couple of really critical things to know, and I want to bring this up for those of you doing cell biology in general. First is, our mechanism of coupling is elastic coupling. But in that very tight space, if I take a sphere here, and this fluid around it, and I have a sphere here, this sphere will influence the other one, viscously. There could be viscous coupling. There's also sort of an emerging area called poroelasticity. That is what happens when you have porous fluid dynamic interactions in elastic structures, like a dynamically squeezing sponge. This is all good news. Here's the bad news. In fluid dynamics, there are these limiting equation systems. One is the Euler equations, which are, say, inertia is all that matters. The other are the Stokes equations, which say viscosity is the only thing that matters. In both of those cases, we assume that the fluid behaves as a continuum. That is, everything is differentiable, everything is continuous. That is true at a spatial scale where your spatial scale of the problem is large relative to the mean free path of molecules involved. This is measured by something called the Knudsen number. The spatial scale of protein-protein interactions is actually violating, it's at Knudsen numbers, which suggests that many of the assumptions of traditional continuum fluid mechanics may be breaking down. And we know this got to be the case for when a protein docks into another one. At that point, if this was a viscous flow problem, the forces go to infinity, mathematically, to do this. But we know that proteins don't generate infinite forces. At some Debye length, the water molecule is just now an item. It's no longer a shearing factor. So this is right in this intervening space, and this is what I call the physics of intracellular worlds, of not quite continuum, not quite quantum. There's no better word than quantinuum mechanics. We don't know how, we really don't have good methods for dealing with this. And so, there.
Basically, you are talking about the incompressible
Right.
rarefied flow in that sense, from gas dynamics analogy.
That's right. Or granular flows.
It's not granular to me. Granular can be totally differentiable. That's different. You are talking about the case that the mean free path and your spacing basically they're comparable, right, in your case.
Right.
So you have this slipping, no slipping condition not applicable. So I think one should not apply the Stokes analogy to this.
I agree.
Okay, so there are some micro nano fluid mechanics studies. They can be, I think they can be helpful.
I agree. I will take them. Whoever has them. Yeah. That's great. So this is the place where, in fact, if the molecules are spaced appropriately, you can't assume certain limiting conditions of the continuum equation. But I would maintain in problems like mitosis, where filaments are pulling chromosomes, there are lots and lots of neat examples. Cilia, flagella, all of these are happening at these very tiny spatial scales where water, its role has been somewhat ignored.
Any other questions? Please.
Excuse me, because you are measuring a mouse method, I'm not sure that is similar to the frog method that I'm quite familiar with. Because in the muscle cell, not only the fibers, the proteins are quite specialized as a range that you introduced, but also the endoplastic reticulum and the T-tubules. These structures bundle these structures like the thin and the thick filament. So could that be the reason that your lattice keep in a range?
Yeah, so the question is that moth muscle isn't like frog muscle. Actually, it turns out it's stunningly like frog muscle. They also have sarcoplasmic reticulum, T-tubules, everything. Now, the interesting thing is as the cell shortens, one could argue that material in the T-tubules is free to go in and out. One could argue that. you're still stuck with the intracellular environment, not free to change volume. And the experiment there, by the way, that data set was frog muscle, the last one, high-speed X-ray. So we're rather indifferent whether it's frog or insect. We use frog if we need what's called constant or tetanic contraction. We use insect if we need phased lock. They're quite similar.
Because the T-tubules and the cytoplasmic reticulum, it like kind of bonding the
Right.
fibers. It's like some ropes that tighten it. So, I mean, could that be the reason that it maintains the distance between the myosins?
Yeah, that's so the student is pointing out, or faculty, whatever, this PhD student is pointing out, postdoc, faculty, president, that the myofibrils are surrounded by what's called sarcoplasmic reticulum, the ER equivalent in muscle. And could this be bracing the lattice? It's possible. I mean, our data suggests that the lattice is indeed undergoing these radial changes. And in a fiber that we never activated, we just did length changes, we see pretty profound expansion and contraction of the lattice. The SR doesn't look like it's bracing it, but I really appreciate that. I think it's
Any other questions?
a half-baked talk, so.
But where there's APTP, there's likely to be magnesium. So have you thought about metal ions as well as just water as a problem, either for your breathing problem or other aspects of your dynamics?
No, we have not. I mean, yes, you're absolutely right. Because when we do our skin fiber preparations, both in insect and in frog, we use MgATP, magnesium-based ATP. And I do know that we have to wait a while for everything to kind of come to a reasonable equilibrium. No, we have not really considered, basically we haven't considered much of anything other than ATP, and we haven't even had a chance. We would love to measure ATP dynamics going in and out.
Questions?
So basically, kind of following up on Professor Shi's question, you used a spring system, just purely springs, which means that it's totally independent of speed. So why is it that you cannot put a dashboard into it and make a traditional mechanical spring dashboard system so that we can take care of the speed issue as well? So there is viscous dissipation.
You can. And in fact,
Is there any problem with that? Or?
no. And in fact, in the early models, particularly the linear ones, we just threw dashpots in. We didn't have a lot of estimates of what they would be. But you can add them. It doesn't give you viscous coupling between individuals based on sort of Stokes flow. But it would give you damping, obviously.
Does that give a better result than what you have, or is that not as good?
It would be very challenging. It would be a challenging addition to Huxley's data on these rapid perturbations. I'd have to think about that. And there's a second question.
There's a second question. You assume that it's the force was proportional to the square root of length.
No, I simply said that the radial dimension has to increase as the length, as the square root of length, not the force.
Oh, I see. Okay.
So as you it's a constant volume assumption of a cylinder, basically. Yeah.
Any other questions? So I have one question is that, as you mentioned to the students, that biology is actually based on evolution. But evolution is not really finding the optimal solution. Evolution is to the for survivals. So I'm more interested on this energy dissipations, because muscle movement is one of the area where a lot of energy on. And you mentioned about the contraction which allow this, we call facilitated diffusions, expansions. And we know the hydrogen pumping can produce ATP. I guess there's no way that through mechanical contraction or expansion, you could also produce those ATP doing this. I'm just, no.
I know what you're asking. Is your question, does that, in that energy of
Because
the,
there seems to be a lot of energy. How nature can capture those energy and those mechanical use.
I understand. If that was rampant speculation, this is going to be even worse. So let me, I have no idea is my first answer. Let me speculate. When you exercise, you get hot. The reason you get hot is your muscle efficiency is low. In fact, the heat is being dissipated by muscle. Where that heat and how that heat is dissipated, what that inefficiency is, partly in the hydrolysis inefficiency. But it could also be in this mechanical shearing process that Professor Shi was suggesting. That can be heating up muscle. Generally, heat dissipation is not, when you're heating up, that's energy you're not using to produce force. The flip side is the warmer you are, the more effective your protein, so your protein reaction rates can be temperature dependent. So I guess one could argue indirectly, so I told you there's rampant, that the heat generation can be used in a more favorable timing of muscle. That was a stretch.
Any more questions from the students? Code 1, Code 2, Code 3? Please join me to thank Professor Tom Daniel for this wonderful presentation.